Hello, everybody. This is just a quick tutorial on the primer mapping activity in our week 6 lab. Remember that the overall goal of this activity is really just to see whether or not the primers that you're using have a good shot at binding and amplifying your target organism's DNA. So the first thing you're going to want to do in this activity is download the APE software. There's a link to that APE software in the lab manual. You can either click on this link here. Additionally, there's a link under the week 6 module where you can just download the latest version of APE to your laptop. So I'm going to go ahead, open up that link. Here you can see that I'm working on a Mac, so I'm going to download the Mac version. If you're working on a PC, you want to download the Windows version. I'm going to go ahead and hit download. Once that software is downloaded, I'm going to go ahead and open it up. If you're on a Mac, you'll see a screen like this, and I'm going to go ahead and drag and drop that into the applications folder to open up the software. If you're working on a PC, you'll have to click on the file to open it up and download it onto your PC. Once it's installed in applications, you can click on the applications button and you'll see the application there under applications. You just want to make sure that you're opening up APE under the applications tab. So I'm going to go ahead, open that up. It's going to say, you're sure you want to open it? Do you trust it? I'm going to hit open, and you should see a screen that looks like this. So the next thing we need to do is figure out what is the genus and species name of the target DNA or the target organism that we're going to find. So just as a simple example, I'm going to say that we're looking for beef contamination in vegetarian burgers. So I need to know the genus and species name of beef or cows. So I'm going to go to the internet. I'm just going to search genus, species, name, cow. And you can see that domestic cattle belong to the genus Bos and the species Taurus. So the genus name or genus species name is Bos taurus. So I'm going to go ahead and write that down in my notebook or write it down somewhere and make sure that I have it written down for the rest of this activity. So the next part of the activity, if you follow in the lab manual, is you navigate to this NCBI GenBank homepage. So I'm going to go ahead and open up this link here. Alternatively, you can just Google NCBI GenBank. You'll see a screen that looks something like this, and you want to make sure that you have nucleotides selected from the drop down here. Now, once you have that set up, you're going to follow the instructions in the lab manual that essentially tell you to look for the target region of the area that you're amplifying in the species or species that you think you're going to detect. So in this case, I'm looking at a vertebrate animal. So I'm hoping to find beef or Bos taurus or a cow. And so the primers that I'm using to detect a vertebrate animal are these primer sets in this column here. So I'm using the COX-1 or mitochondrial COX-1 DNA, or that region of the cow genome or any vertebrate genome. That's the region that I'm amplifying. And so I need to search for that particular region because that's the region that the primers that I'm using are targeting. You'll use a different region if you're looking for plants. You'll be using the RBCL primers with search within the chloroplast genome. If you're looking for fungi or algae, you're going to be looking for the ITS region or the internal transcribed spacer region. So given that, the first thing that I'm going to do is go into the NCBI database, and I'm going to use these search terms because this is the primer set that I'm using. So I'm going to look up the complete species name, Bos taurus. So that's the cow, and then I'm going to put complete, and then I'm going to put COX-1, and I'm going to hit search, and then we'll see what comes up. So here I've got a bunch of weird stuff coming up that are not the species names. You can see a bunch of different hits. So I'm going to narrow it down to just the Bos taurus hits. So if I click over here, it says Bos taurus, and I'm going to click on this link right here to narrow it down to just Bos taurus. And here you can see a bunch of hits that say the genus and species name Bos taurus, and then you can see it says mitochondrion, and then it says complete mitochondrion genome. So this will work really well. It's about 16,000 base pairs long, and this will include the region of DNA that I'm interested in amplifying. So I'm going to go ahead and click the FASTA part right here. And here you can see the entire sequence of an individual cow that had its mitochondrial DNA sequenced. And this is 16,000 base pairs long. So I'm going to copy just this region. Here you can see that there's a little bit of ancillary information. So this is the accession number. So this is the individual sort of cow that had its mitochondrial DNA sequenced. Here's the species name of the species that was sampled. Here's an isolate number. And then it tells you that this is the complete genome of mitochondria sampled from this cow. And so I'm just going to highlight the actual species information and not that ancillary information. I'm going to copy that, and then I'm going to paste it into this APE window. And here you see something like this. So this is basically telling you, here is my entire string of DNA, and each of these numbers marks a region in that sequence of DNA that we have. So this is at 10 base pairs, 20 base pairs, 30 base pairs, all the way on through to 16,440 or so base pairs. And you can see that we have the entire sequence length listed right here at 16,448, which matches what was listed earlier, so that's a good thing. So the next thing we have to do is figure out whether or not the primers that we're using are going to align in this sequence. In other words, are they going to anneal somewhere in the region of this target species that we're looking for? So we've got to go and look at the actual sequence of the primers that we have. You can find that in your lab manual. What I recommend is just copying and pasting from the electronic version of the lab manual. So I'm going to go up here, and I'm going to find the primer set that I used for my target organism. So in this case, I'm looking for a vertebrate. So I used these vertebrate primers. If you're looking at a different taxonomic group, obviously you should use the primers that align or that you used on your PCR this week with your target organism. So the first thing I'm going to do is copy and paste the forward primer, and I'm highlighting just the nucleotide sequences, not the 5' or 3' end. So I'm going to go ahead and hit copy. And then what I'm going to do is use this software to search within the cow mitochondrial genome to see whether or not those primers align anywhere within this target area of the target taxonomic organism I'm looking for. So the way you do that is you can either do edit and then hit find like this, or you can hit Command F and get that screen up, or there's this little icon here that looks like a magnifying glass, and you can hit that. And what I'm going to do is just paste my forward sequence into here. And essentially what I'm telling the software to do is, can you find a region where these primers will complementary base pair to the DNA of the cow here? So I'm going to go ahead and make sure I select also find reversed string, and then I'm going to hit find next. And what you can see is it says no match found. And that's because I'm saying, well, only allow it if it's a perfect match, but you can up the number of mismatches that you can allow in the find. So now I'm going to search under, see if you can find somewhere where this matches where there's one mismatch, and hit find next. It still says no match found. Now I'm going to up it to 2 and hit find next. And look, now it said found forward match at region 5,709. And so basically there's a spot where that primer matches where there's only 2 mismatches between my sequence and the target DNA. And so what that means is there's a pretty good probability that this primer set is going to be able to anneal to my target DNA. So what I'm going to do next is hit close, and then I'm going to go up to this features button, and I'm going to click new feature, and then I'm just going to call this F primer for forward primer. You can name it really whatever you want. And then I'm going to hit OK. And what you can see now is it's added this little forward primer area to the sequence. So first important piece of information checked off, there's a spot where my forward primer anneals within the target sequence DNA. So next I'm going to look at the reverse primer and see whether or not the reverse primer also anneals. So I'm going to copy the reverse primer from here. I'm going to go ahead and hit find again. And this time I'm going to paste the reverse primer. And I'm going to hit find next. And you can see that the reverse primer also matches at 6,392 base pairs at this portion of the region, so downstream from where the forward primer is, and it matches with 2 mismatches. So once again, it's not a perfect match, but it's few enough mismatches that we can feel pretty confident that this primer set is going to work, this primer set is going to anneal, and the PCR is going to work. So I'm going to hit close again while this is all still highlighted. This region is all still highlighted. I'm going to hit the features button. I'm going to hit new feature. I'm going to put R primer for reverse primer. And then I'm going to go ahead and hit this little reverse com button just so it knows to basically link it in the reverse complementary sequence. And then I'm going to hit OK. And now we've got the reverse primer added on as well. And so here we can see the forward primer anneals pretty well with only 2 mismatches. The reverse primer anneals pretty well with only 2 mismatches. And then what we can do is we can actually highlight the DNA sequence from the forward primer all the way to the reverse primer and all the DNA sequence that's in between. And so this is essentially telling us the region of the DNA that our primers are going to amplify within the target species that we're looking for. So here it basically tells us that the length of the amplicon that we're going to amplify is 709 base pairs long. So from the back end of the forward primer, the front end of the forward primer to the back end of the reverse primer, this sequence or amplicon is going to be 709 base pairs long. And so what you can do is take a screenshot of this sequence or this region. So take a screenshot of all of that. And then you can copy and paste that into your lab notebook and answer the appropriate questions in your lab notebook associated with where your primers are binding and the total length of your amplicon.